All right, good morning, good afternoon, and good evening, everybody. My name is Abby Kropelnicki, and I will be your host today. Thank you so much for joining us for this exclusive webinar, Next-Gen Metabolomics, Absolute Quantification with the Giga System, jointly hosted by the TMIC Wishart Node and Metabolomics Inc. TMIC is a nonprofit core facility at the University of Alberta based in Edmonton, founded by Dr. Wishart in 2011. It offers cutting-edge, targeted quantitative metabolomics assays, and it supports academic research with advanced instrumentation and world-leading metabolomics software and databases. And on your right, Metabolomics, also known as MTX, is a spin-off from the TMIC Wishart node launched in 2019 and is based in Edmonton, focused on producing and distributing quantitative metabolomics kits worldwide. All right, so for today's session, it will be led by two distinguished experts, Dr. David Wishart and Dr. Alan Zhang. All right. We will also be having a Q&A session after the presentations, so feel free to submit your questions throughout the webinar via the Q&A button at the bottom of the screen, which is being circled in red currently. However, your questions will be answered during the Q&A session near the end. Also, please note that that chat function has been disabled for this webinar. All right, and finally, a really exciting feature of this webinar, we're going to hold a giveaway draw for a free GigaKit analysis of 82 samples by our TMIC experts. This prize is worth up to $25,000, so you're definitely going to want to stay till the end to claim that prize. You must be present to claim that prize, and please note that this offer is valid up to 6 months, and all samples, plasma, and serum need to be sent in one batch. All right, so please stay till the end, and that will be the giveaway draw. So let's move on to our panelists today, Dr. David Wishart. So it's my pleasure to introduce him today as a distinguished university professor at the University of Alberta and a Tier 1 Canada Research Chair in computational metabolomics. A pioneer in the field, he co-founded the first metabolomics company and led the Human Metabolome Project and database. Dr. Wishart has developed numerous widely used tools, including DrugBank, HMDB, MetaboAnalyst, and FoodDB, supporting researchers all around the world. His current work combines AI, robotics, and advanced biosensors to drive innovation in health, agriculture, and environmental science. He is widely recognized as one of the most influential biological scientists of our time. Please welcome Dr. David Wishart.
Thanks very much, Abby, and thanks again for that introduction. I'm just going to share my screen here. So I think we're A-OK on the screen sharing.
Yeah, that's perfect. Thank you.
So it's my pleasure today to introduce what we're calling the Giga System. And we're calling it a form of next-generation metabolomics, sort of in line with next-generation sequencing. And we think it's an important development in the field of metabolomics. I want to first give you a little bit of a background or motivation for developing the Giga System. I think many of the people here at today's webinar are aware of untargeted and targeted metabolomics. In the case of untargeted metabolomics, many of us are attracted by the very large coverage that's possible. Typically today, most untargeted methods allow you to identify more than 500 compounds at a time, and some of the top labs can measure more than 1,500 compounds. And that's very appealing, and in fact, that coverage for untargeted metabolomics is what's made it so popular. But it's important to remember that untargeted assays are not quantitative. You don't get concentrations out of them. It's also important to remember that untargeted methods are not very standardized and therefore not very reproducible. Every lab has their own method. Every lab has their own system. Every lab produces a very different list of compounds for exactly the same samples. That's a problem. It's also an issue that even though data collection for untargeted metabolomics can be quite quick, often less than 20 minutes for a single sample, the data processing and data analysis can take hours, sometimes even days per sample. That really slows the process down. So there are pros and cons for untargeted metabolomics. Now, on the other hand, targeted metabolomics, which is not as common, offers a quantitative approach to measuring metabolites. Now, typically, one of the cons about metabolomics is you don't measure as many compounds, typically fewer than 200 with most of the commercial assays that are available today. Some of the better commercial assays or in-house assays can measure more than 500 compounds. But the central advantage to targeted assays is they're fast, they're standardized, and they can also be easily automated. Typically, data processing times are 1/10 to 1/100th the amount of time for untargeted metabolomics methods. Quantitation is really key, and this is one of the things I want to emphasize here. If you want to have a standard system, you want to have standard quantification. Everyone understands what micromole and millimole are. So quantification is essential to standardization. It's key to reproducibility. It's essential to getting high quality results. It's important for traceability. If you want to translate your findings to the clinic, you need quantification. If you want to translate your findings to drug industry, you need that for FDA approvals. If you're doing forensic testing, if you're following any kind of government regulated testing or environmental testing, things have to be absolutely quantitative. Likewise, if you want to have your assays approved for ISO 17025 or 15189, or for CLIA or COLA compliance, they have to be quantitative. So what we were looking for is just essentially trying to find a quantitative metabolomic assay that could actually exceed that 1500 limit of what we see with untargeted metabolomics, but also create a system that would be just as fast, just as easy, just as standard, and just as ISO compliant as a fully quantitative targeted assay. So can you blend the best of untargeted with the best of targeted? And the answer is yes. And that's why today we're introducing the Giga System. That includes both an assay that can be run locally here in Canada through TMIC, or the Giga Kit, which you can acquire or purchase from Metabolomics. And I'll explain these a little bit more in detail, and we'll follow on with more descriptions of the Giga Assay and Kit with other videos and slides. So when we were designing the Giga System, we wanted to make it into a 96-well format. And this is a 96-well plate that allows you to do automation. Many systems, whether it's HPLC, robotic systems, LC systems, MS systems, support 96-well plates. We also wanted to make a very simple workflow, something that anyone could do. We wanted to make it something that could be kittable, something that could be put into boxes. All the materials, reagents, plasticware could be put into 1 or 2 boxes and make that easily shippable. We also wanted to make sure the assay was sensitive enough that you could work with less than a drop of blood, about 50 microliters. We wanted to have just 30 minutes or less for the LC MS runs for each sample. We wanted very broad coverage of metabolites, many classes covering organic acids, amino acids, nucleotides, indole, sugars, lipids, and so on. And when we put the list together, we realized it was going to be probably more than 1,600 compounds. Now, the advantage when you can do absolute quantitation, it means you can add numbers up, or you can calculate ratios. So we call these sums and ratios. And these are essentially bonus analytes, and allows you to go far beyond just the 1,600 individual compounds to measure to maybe up to 2,500. To make the system fully quantitative, we wanted to have multi-point calibration. We wanted to have hundreds of isotopic standards. We wanted to have great inter-day quality control accuracy and recovery. We wanted very low coefficients of variation, under 15%, and very low limits of detection, under 1 micromolar, and low limits of quantitation for most of the compounds. We also wanted to make the system compatible with multiple LC-MS platforms that we have in our own labs, but as well with other MS platforms that are in many other labs. And not only in terms of the data acquisition, we wanted to make data processing fast. We wanted to change the amount of time from days or weeks to analyze a single sample to less than 20 seconds per sample, and offer software that could do this through the web, making sure that that software is accessible to anyone on any platform. So the result is we've produced 3 things. One, we've been able to produce kits that are shippable. There's a picture of one of the kits, or the Giga Kit. We wanted to create the assays, which basically use the kits, but we allow us to run them in our own facility here in Edmonton. And we also produced robotic systems that allow us to not only make the kits, but also to run the kits automatically. Now, in developing the system, not only the assay and the kits, we wanted to make things inexpensive. And to do that, we've done something called chemical tagging. So this allows us to do a number of things. It allows us to run on a single LC column instead of multiple columns, even though we're still measuring almost 1,800 compounds. We wanted to make the system compatible with reverse phase because we get the best separation through reverse phase. We wanted to use tagging to enhance ionization efficiency, which in fact allows us to measure things with up to 100 times greater sensitivity if it was not labeled or tagged. And this also greatly reduces the cost of the isotopic standards and the number of isotopic standards we have to use. To enhance the coverage to get up to around 2,000 compounds, we have to run 2 different LC runs, one for the positive mode, one for negative mode. And we also use flow injection analysis for lipids and acyl carnitines. To make the quantitation as accurate as possible, we've done 7-point calibration for all the liquid chromatography detected molecules, and we use single-point calibration for the FIA detected molecules. To make the data processing as fast as possible and to make the system as portable as possible, we have pre-calculated MRM transitions that can also be easily ported or transferred. And we have retention times also that can be easily ported and transferred into your system if you're running the kit. To make the sample prep as fast, simple, and as multiplexed as possible, we've got 2 types of 96-well plates that are used both in the assay and the kit. And again, that greatly simplifies the number of pipetting steps. To make it as simple as possible, we've prepared everything as pre-mixed standards and plates, things are pre-weighed, everything is pre-packaged and pre-prepared. And critical to all of this, especially with this large number of analytes being measured in each sample, almost 2,000, we had to develop software, and we've used AI-enabled software that allows us to do up to 200,000 analyte measurements in about 10 minutes. Now, this is not our first trip to the rodeo. As some of you might know, we've actually been developing and distributing and offering assays and kits for other types of assays. The first kit and assay we developed internally was called TMIC Prime, and that came out in about 2019. And then in 2023, we produced what's called TMIC Mega. The Prime measures about 145 metabolites, the Mega up to about 720. They use dozens of isotopic standards. They are ISO compliant and ISO approved. They can work for plasma, urine, and stool samples. They're compatible with 96-well plates. They can work with as little as 40 microliters of serum or 20 microliters of urine. And we also made these systems compatible with all of the triple quads and high resolution systems in our lab, as well as many other ones that are available commercially. The workflow for these kits is highlighted above in the slide, and we can show how things are used in different plates. Isotopic standards are processed, things are derivatized, sent through the mass spectrometer, and then processed using the LC autofill software. Now, the challenge was that the Prime and Mega certainly showed us how to make reproducible, fully quantitative targeted assays and kits. We learned how to make or get by with minimal volume. We learned how to run them and design them so they could be run by anyone and shipped anywhere. But the limitation was that 720 metabolites, that was not comprehensive enough to cover all of the major metabolic pathways and chemical classes. And so it's been a journey up a hill where we started in 2019 with 145, 2023 with 720, and today almost 2,000 analytes with the Giga system. And I think it's reached a stage where we can be, at least, maybe we haven't climbed Everest, but we've certainly climbed a very tall mountain, making quantitative metabolomics as comprehensive now as the best untargeted assays. So the Giga system is able to measure 1,780 distinct metabolites, and because these are quantitatively measured, it means we can calculate more than 700 different sums and ratios. This works for serum and plasma for any mammalian system. We cover 38 chemical classes. We can work with as little as 40 microliters of serum and plasma. The compounds we chose and specially tested for are of particular interest for biomarkers, for clinical research, biomarker discovery. We're just wrapping up the systems for urine and stool samples, and that will come in another month or month and a half. Of course, it's been adapted to 96-well formats as our other systems like Mega and Prime. And as I said, it's also being run robotically in our labs currently. We have validated it on Agilent and SciEx platforms, and further validation on Waters, Thermo, and Shimadzu instruments are coming in the next month or two. So we have both an assay and a kit. This just highlights the assays and kits that are currently supported. So SciEx 5500 and 6500 triple quads, Agilent 6495D already out there. SciEx 7500, Waters Zevo Absolute, Thermo Orbitraps, Thermo Altus, Thermo Explorer systems, and Shimadzu triple quads will all be wrapped up by the end of the summer. And so all of these platforms will be supported with both the Giga assays and the Giga kits. As I said, we've been working with and doing robotic automation, not only for the preparation of the kits, but even running the kits. We have a pipetting and liquid handling system that's going through the final rounds of testing and use in our labs. We know that when people have to do pipetting and liquid handling, especially with 96 wells, doing it manually, it can be prone to errors. It's tiring and manually intensive. We know as well that people have to be trained, and you need to be fully engaged with good concentration to get the high quality results. What we'll be releasing shortly is an ultra low cost robotic system that allows you to run the Giga kits almost entirely. The concept of just lock, load, and leave. And this is just an image of the robotic system that we're currently in the final stages of testing for this. So what we're trying to do with the Giga system is give you choice. You can either run the assays if you are not a mass spec lab, or if you have mass spec equipment, you can run them as kits. The prices here are listed in Canadian dollars. So if you wanted to convert those to US dollars, you multiply these by 0.7. So in the case of the assays, the price range is between $200 to $300 per sample. In the case of the kits, the range between 120 to 145 Canadian dollars per sample. Again, multiplied by 0.7 if you want to convert those to US dollars. Now there's a price range for a variety of reasons. Prices vary based on the number of samples that you might submit for the assays. So a very small number of samples is charged more. A larger number of samples is charged less. It also varies with the type of instrument that you're running on. Some instruments are less sensitive or slower in operation, so that also changes the price. It also depends on the amount of data analysis you're requesting or the amount of bioinformatics analysis that you're requesting, again, for the assay. That will change with price. It also depends on the wait time. So if you want immediate turnaround, prices are higher. If you can wait 4 to 6 weeks, the prices are lower. The kits, there's a little bit less variation, but again, that's also a function of instruments and other differentials. And of course, with the tariff wars going around, that also affects some of the pricing for these systems. It's important to note that when you get up to very large numbers of samples, a very large number of kits, very significant discounts are available. And then as part of our mandate, because we're funded through the Government of Canada, we offer very special discounted pricing for labs in developing countries in our effort to democratize access to metabolomic systems. There's lots of applications for Giga now that we've got this sort of threshold of around 2,000 compounds. It's a great vehicle for biomarker discovery, and I'll show you an example. It allows obvious very rapid translation because you're doing quantification, and it does do general research in metabolomics. It's also a great way for people that are interested in clinical or drug applications to learn how to develop, assess, and validate locally or lab developed tests, LDTs. With so many organic acids that are measurable through the Giga system, you can detect and monitor more than 100 different inborn areas of metabolism, and you can do this at a fraction of the cost and with very minimal instrument requirements or time. We've ensured through our selection of the metabolites in this system that it covers many or most of the metabolic and clinical chemistry markers that are known in clinical labs or which have been published over the last 10 years. It's possible to get reference values and reference cutoffs for all of the biosamples that we typically can measure. And this is also available through the LC AutoFit software that comes with the kits. The Giga system is flexible, so you can expand it. We can customize it by adding and removing up to 24 different metabolites for different applications. And it's something that now that we know how to do this, can be expanded significantly more over the coming months and years. I said we would show you an example of one of the applications. And this is an example of cervical cancer and trying to use a simple blood test, to say basically a drop of blood, to identify people who have early stage cervical cancer. In the case of North America, cervical cancer is one of the fastest growing cancers among women. And so in order to look into this particular one, we had a study where we looked at about 20 individuals in early stage cervical cancer and another cohort of about 20 or 25 individuals who were healthy controls to see if we could identify biomarkers for early stage cervical cancer detection. Now, the issue here is that there haven't been good biomarkers, and in part it's probably been due to the limited coverage of metabolites and the fact that many of these things were not quantitatively measured. So the test involved collecting blood samples, purifying the plasma, running it through the Giga system, and then processing the data with LC AutoFit and analyzing the numbers using MetaboAnalyst. I'm showing here results of this. We did both simple t-tests and volcano plots. And if you look at the upper reaches of both plots, you'll see certain characteristic features. So a number of organic acids that are significantly reduced, like methyl-adipic acid, pamelic acid, azelaic acid, capric acid, and then other compounds, particularly phosphatidylserines, greatly increased, particularly those on the right on the volcano plot. These were surprises in part because these metabolites were not and are not measurable in things like the mega or the prime assays or other commercial targeted assays that are typically used today. On the left, I'm showing the PLSD plot where we can see the clear separation between the cervical cancer cases in red and the controls in green. In the middle, we're just showing the VIP plot, and you can see quite a number of metabolites that have scores above 2.0. And the ones that are showing up, again, are phosphatidylserines, purines and nucleotides, and as well as a number of fatty acids. What was striking and particularly novel was the significant increase in a number of phosphatidylserine levels. These are not measured in most assays, and this is probably one reason why this particular set of markers had never been detected before. The decrease in certain fatty acids also was unexpected. Again, it's because many of these things are not measured in most available targeted assays. So this is giving us new insights, new biomarkers, and helping us understand some of the metabolic pre-programming and gut dysbiosis issues that might be associated with cervical cancer. So to wrap up here, I really want to emphasize that what we reached in this stage, this age, is a shift, a sea change, that quantitative metabolomics, which has sort of been the finishing last in these races in terms of compound coverage, now is surpassing the coverage of the best untargeted assays out there. It exceeds what can be measured with Metabolon. It exceeds what can be measured by the best untargeted assays from the most advanced labs. And instead of just getting compounds, you're actually getting absolute quantification. In addition, quantitative metabolomics is fast. Sample processing times of minutes, data analysis seconds. It's kitified, making it easy. It's very robust. It's fully standardizable. with the robotic systems, it's fully automatable. These are now ISO compliant, they're CLIA and COLA compatible, and they're adherent with FDA requirements. With the robotic systems that are going to be coming out this summer, it'll make the Giga system even cheaper, it'll make it even faster, and will make results more consistent. What we've also done in trying to create this Giga system is we're giving you choice. You can either run the assays in a central lab, such as TMIC, or you can run your own kits and assays. Ultimately, the choice is up to you. So a Giga system could not have been developed without the effort of a number of key people. And you'll hear from Alan shortly, who is the key developer, but I want to also thank Rupa, Tammy, Yamile, Sukanta, Siang, Scott, and James for all their work in designing, testing, and validating the Giga kit, the Giga assay, and the robotic systems. Certainly, if you have questions about the Giga system, I'd encourage you to contact Rupa Mandel, and her email is given here. So if you want to write it down, I'll keep the slide up a little bit longer. And she can provide you with a lot more information about the details for Giga. We're going to give you a little bit more information in the coming few minutes of the presentation. And what I'll be doing is following on with a brief video that gives you a bit more detail. And then Alan will give you a follow-on slide presentation going into much more detail about the specific compounds and processes to run the Giga system.
All right, thank you so much, Dr. Wishart, for that insightful presentation. It's very exciting, and we hope that made things clearer for the audience. But of course, if anyone has questions, we do have that Q&A box at the bottom, so feel free to type your questions and we'll answer them at the end. But yeah, as Dr. Wishart transfers over with his video, I just think that it's going to be a very transformative kit. It's going to accelerate discoveries, streamline workflows, ultimately open new doors for researchers around the world. So it's very exciting. I think that Dr. Wishart's ready. Are you ready, Dr. Wishart?
Yeah, so I think we'll just play the video now.
Welcome to the future of quantitative metabolomics. Introducing the Giga System, a next-generation solution enabling absolute quantification of small molecules with unparalleled coverage, efficiency, and precision. With just a single drop of blood, the Giga System can quantify nearly 2,000 metabolites and 700 sums and ratios, spanning 38 distinct chemical families. This includes lipids, amino acids, organic acids, and many others. With such comprehensive metabolomic profiling, the challenge becomes efficient, reproducible data processing, a challenge we meet with LC AutoFit. LC AutoFit is an automated data processing program that you can access online from anywhere, and an offline version is coming soon. Simply register your kit, upload raw data, run the analysis, and download fully quantified results in minutes. An intelligent flagging system highlights potential issues like inconsistent retention times or low signals, helping researchers quickly identify and resolve problems. Automated retention time recognition minimizes manual input and ensures consistent alignment across runs. Our Giga system supports multiple MS platforms, including SciX QTrap and Agilent QQQ, with new instrument compatibility continuously expanding. To further streamline the metabolomics workflow, we're addressing the challenges of manual pipetting, which can be physically exhausting and prone to errors that jeopardize results. That's why we're developing a robotic auto pipette that automates much of the sample handling process, ensuring consistent, precise, and reproducible pipetting every time. Designed for ease of use and minimal training, this system reduces strain and frees researchers to focus on data analysis and scientific discovery. Thank you for joining us as we unleash the promise of next-generation metabolomics.
All right, thanks so much for that video, Dr. Wishart. We're going to move forward now to Dr. Alan Zhang. He's going to be presenting a deeper dive into the Giga system, but first I'm going to introduce Dr. Alan Zhang. He leads the development of metabolomics assay platforms and supporting software at MTX and the TMIC Wishart node. His work focuses on advancing high-throughput, quantitative metabolomics technologies and enabling scalable, automated data analysis solutions for clinical and translational research. Take it away, Alan.
Thank you very much, Abby. I'm going to share my screen here. So hello, everyone, and thank you for joining us today. I'm very excited to introduce our latest innovation in targeted metabolomics, the Giga System, a next-generation platform designed to transform how we generate, scale, and apply metabolomics data. You've seen the big picture from the video previously. Now let's take a closer look at what makes the Giga System unique. The Giga System is our most advanced targeted metabolomics platform, designed for high-throughput, fully quantitative analysis. It quantifies up to 1780 distinct metabolites and over 700 biologically relevant ratios, covering 38 chemical classes. The platform incorporates nearly 300 isotope label internal standards, ensuring accurate, reproducible quantification across a wide range of metabolite types, many of which are clinically significant. The system is optimized for 96 well-play workflows, uses just 40 microliters of biofluid, and is fully compatible with automation and the robotic systems. The Giga System is also ISO certified. It has been validated on SciEx and Agilent triple quad instruments, with additional mass spectrometry platforms coming soon. Validation protocols for urine and fecal samples are also currently in progress, and it will be released later this year. Let's take a closer look at what the Giga System actually covers. Let's start it with the chemical diversity of the metabolite it targets. The panel here includes amino acids, organic acids, sugars, bile acids, nucleotides, vitamins, fatty acids, and an extensive set of lipid species like phospholipids, sphingolipids, and isocarnitines. Together, the 38 chemical classes represent a comprehensive cross-section of metabolism, spanning both water-soluble and lipid-soluble metabolites. This level of chemical coverage ensures that Giga System can support diverse research needs, from fundamental biochemistry to disease biomarker discovery. On this slide, we show how the 360 targeted non-lipid small molecule panel is functionally aligned with disease-relevant biology. As you can see here, each row corresponds to a major disease or biological process, such as inflammation, cancer, or diabetes. The number indicates how many metabolites in a panel are associated with that category. For example, there are over 200 small molecules are involved in gut microbiota-related pathways, and more than 150 are tied to cancer and inflammation. This alignment supports not just biomarker discovery, but also mechanism interpretation, which helps you understand how different biological systems shift in different diseases. This slide focuses on the targeted 1,400 plus lipid species, grouped by their biological relevance as well. Many of these lipid groups, like phosphatidylserines, isocarnitines, they are clinically relevant and often overlooked in different studies. By offering structured coverage across both small molecules and lipids, we believe the Giga System enables a comprehensive yet interpretable view of metabolic dysregulation in health and diseases. As shown in the diagram here, the Giga System workflow is based on two pre-analysis sample preparation panels and runs with automated data processing via LC Artifice software. The goal was simple: eliminate complexity without sacrificing performance. So the workflow was developed not only for accuracy, but also for scalability, especially in labs running dozens of hundreds of samples per week. As you can tell here, what was once a complex, heavily expertise-dependent LC-MS process has now become fast, reliable, and user-friendly. Let's take a closer look into the sample preparation workflow. For panel A with DFI, The sample preparation time is about 3.5 hours for 96 samples. Only roughly 2 minutes per sample, which is quite efficient for targeted workflows. The process begins with sample loading and dermatization, followed by drying under nitrogen flow. And after extraction and filtration, samples are ready for LC-MS analysis. For panel B, it's even faster, around 3 hours per plate for 96 samples. The process involves samples and reagent loading, followed by an hour incubation under 60 degrees Celsius. After adding internal standards, diluting, and shaking, the plate is also ready for LC-MS analysis. The protocols are compatible with robotic systems, allowing seamless integration into high-throughput labs, whether they are academic, clinical, or commercial. We also want to highlight the performance quantitative reliability. Across 3 days, 3 concentration levels, and multiple runs, the Giga System maintained an inter-day QC accuracy about 101%, with the low RSD of just under 5%. Recovery was also excellent here, with an average of 98% with tight variation. These metrics ensure you're working with data you can trust, not just for relative comparisons, but for longitudinal and clinical grade studies as well. In short, the Giga System delivers both breadth and precision, which is often a trade-off in metabolomic studies, but here they can come together. Platform compatibility is also a key consideration for us. The Giga System is validated on multiple mass spectrometry systems, including SciEx and Agilent triple quads. We're continuously expanding support to include more platforms like Waters' Ex Evo Absolute, Thermal, and Shimazu MS systems, based on demand and validation timelines. This flexibility means you're not locked into a single vendor anymore and can integrate the Giga System into existing analytical workflows with minimal disruption. Another critical component of the Giga System workflow is the LC Autofis software. It's a fast, intuitive software. It's built specifically for our assays and with 3 access. It takes you from raw data to ready-to-use output in just a few steps. It automates critical steps like retention time recognition, peak picking, and quantification, significantly reduce the time and variability typically associated with LC-MS data analysis. Importantly, LC Autofit is also FAIR compliant, which means your process data is transparent, reusable, and ready for downstream applications. It's currently available online, but an offline version is also in development to meet the needs of users in secure or regulated environments. This is what the process of LC Autofit looks like. Simply register assay plate, upload data, let the system do the work, and you'll get a structured Excel file, which includes metabolite names, concentrations, HMDB IDs, and the reference ranges. The entire process is streamlined and efficient, and is compatible with multiple mass spectrometry platforms at the other part of the system. Now we'll cover the main features of the system. Let's talk about how to get started. We designed Giga to be flexible, as Dr. Wisher mentioned. We know some researchers prefer to outsource analysis, while others prefer full control. Here we support both. We offer 2 options here. Option 1, you can send your samples to the TMIC Wisher node, where we'll run the entire analysis and return a full data report, typically within 4 to 6 weeks, which includes metabolite names, concentration, HMB IDs, LODs, reference ranges, and FAIR compliant output. This is ideal for teams that want fast results without investing in equipment or training. Here is the option 2. You can also purchase the Giga Kit from Metabolomics Inc. and run the workflow in your lab. Each kit comes with everything needed, including buffers, standards, reagents, and protocols. And LC Autofit handles the data processing for free. We want users to be able to focus on discovery, not on troubleshooting methods or sourcing reagents. Kits typically ship within about 2 weeks of ordering. This model is perfect for labs that already run compatible mass spectrometry system and want scale internally. To sum it all up, the Giga System quantifies up to 1780 metabolites and over 700 ratios across 38 chemical classes. It uses nearly 300 isotope labeled internal standard, which is the most comprehensive on the market. It's validated for plasma serum with the version of urine and fecal samples coming soon. Our free accessible software LC AutoFit powers full automation and fair compliant data output. And it's available either as a full service assay or standalone kit. It's flexible, scalable, and also ISO certified. All in all, Giga is built to meet the demand of more than metabolites, from discovery to clinical translation. If you'd like to learn more, get a quote, or start a conversation, please visit our website or reach out by email. For service-related questions, feel free to contact our renowned manager, Rupa Mandel, by email. Thank you for your attention.
So thank you so much, everybody, for connecting with us. We hope that you enjoyed this presentation, and we look forward to connecting with you all soon, and we will try our best to get back to all of your amazing questions today. So thanks so much, and please reach out to us soon. Have a great rest of your day.